And it's a real pleasure to be here today. I've been looking forward to this meeting in Chicago for quite some time since Kara sent the kind invitation. I've had the chance to meet many of you in the community, including dozens of Zoom meetings with several of you. But this is my first chance to interact with a lot of you, an embarrassing large fraction of you, in person. So I'm really grateful for that. And I'm also really grateful for this chance to learn from you, both families and scientists alike, because we as a lab are new to this space. And everything that we've learned here has been from reading papers that have been published or stories that have been shared by folks in this audience over the last few years. So I get to do a slightly repetitive pass over some of the material that Rohan just covered, but with a slightly different hat on as a physician scientist to complement what you just heard about. And I'll also draw out a couple of examples to sort of bring out to highlight in 3 dimensions how some of this ASO development process could work. So to get going, first, my most important slide, if I can figure out how to advance them. Usually I'm reasonably able to do these things, but let's see here. Can I use page down or the arrows or something like that, or just the buttons? No problem. It's a PDF file, if that makes a difference. There we go. Perfect. Great. My first most important slide is actually to show you the pictures of the folks who are actually who have been doing the work over the last several years that I'll be describing to you. In my laboratory in the top row are Tojo Nakayama, a very talented MD PhD physician scientist who was with me for several years. He's now taken an assistant professor position at Tokyo Medical Dental University, but he's done a heroic work, including the bulk of the experiments that I'm going to describe today. Aubrey Susi, a bioinformatician in my group, Emma Sherrill, another assistant bioinformatician who works with Aubrey, Ali Rahman, Tori Sislovich, and Miwa Higashi round out our team. And I also want to gratefully acknowledge up front the collaborators, clinical and scientific, that we've had the pleasure to work with, as well as Rohan and his family, who really, Rohan, you were our introduction to this condition. We learned more about it through folks like Sarah James and Ed Cooper and Ann Perduri, but folks on this slide were essential resources and have been really generous collaborators throughout. Any mistakes we've made are entirely our own, but any successes we've had have been really with the help of the folks on this slide. Since it's my first introduction to many of you, I just want to put this project in a little bit of context. Our laboratory numbers about 20 folks in the Division of Genetics and Genomics at Boston Children's Hospital. And until about 5 years ago, we focused on topics of research on the left, on genome sequencing to understand the basis for neurologic disease, understanding the genetic architecture and the neurobiology of autism. And it's really only in the last 5 years that we pivoted into the space of attempting to do something very challenging, which is to develop therapeutic interventions for genetic diseases. And this all got started, as Rohan mentioned in his slide, with a project that we undertook to initially try to give a diagnosis to a child with a very serious genetic condition who was missing a complete diagnosis. And that diagnostic odyssey actually led us in a really unexpected direction, where we discovered she had a type of mutation that we had a hypothesis about how we could try to fix. And one thing led to another. And over the course of a year, we found ourselves developing a novel therapeutic for this child, for this very rare condition, using antisense oligonucleotides. And that effort has served as the inspiration for several other efforts in our hospital to try to develop antisense oligonucleotides as so-called individualized medicines. I say that so-called because they're not always N's of ones. But we've managed to try this, to accomplish this difficult task of going from bench to a therapeutic that's administered at the bedside in 4 projects involving 6 patients in the last several years. These diseases are shown up here as the vanguard of what we've been able to do, but there have been many other cases in the background that we've been working on just as hard, including KCNQ2, and that's the context that I wanted to share with you. The additional piece is that we're not the only ones doing this type of work these days. That beginning with that first effort in 2018 with Millicent, we've seen many other folks at multiple institutions around the world who have stepped in to develop drugs in this unusual fashion. And it's a difficult space. We're all learning together, but each one of these efforts is hopefully giving us lessons, hopefully successes, but even in those cases where we don't yet have success, we'll get lessons for how to do it better in the future. I'll skip over the N of 1 Collaborative is one of these groups that I'd like to highlight, but in the interest of time. So let's get back to KCNQ2. So again, our entry into this field was recognizing what you all know very well, that there are 2 genetically and clinically distinct forms of KCNQ2, one that's benign, that's caused by heterozygous loss of function mutations, and one that's very severe, a neonatal epileptic encephalopathy that's caused by toxic and interfering dominant negative mutations, usually missense mutations. And it was that recognition that allowed us to develop this therapeutic hypothesis that if we could selectively inhibit the production of the mutant copy of KCNQ2, that we can convert the benign form to we could convert the severe form to the benign form by specifically turning off the poison copy of the gene. To do this, we turn to antisense oligonucleotides, which, as folks know, are a technology that allows you to target any specific gene in the human genome. And by coding by programming the antisense oligonucleotide to match the sequence of your target, you can get it to stick to a location where it mimics a viral intruder to the cell. And the cell then mistakenly recognizes it as something that to be attacked, and it degrades it and then removes it from the circulation. To give an example for how this works, I'd like to share an example that we've talked about actually just about a year and a half ago in Chicago, describing how this could work for another developmental epileptic encephalopathy called KCNQ1 epilepsy. KCNQ1 epilepsy is a condition which can cause a very severe clinical presentation called epilepsy of infancy with migrating focal seizures. Infants born with this really unfortunate condition develop dozens of seizures per day in the first week of life, and they're unresponsive to anti-epileptic medications. And a canonical example of a type of mutation that causes this very, very severe presentation is this R474H mutation in the gene KCNT1. This gene, KCNT1, encodes a potassium channel that's expressed broadly in the brain. It regulates the excitability of neurons and neuronal circuits. And the effect of the mutation is to mutate the channel in such a fashion that it lets in too much current. This is supposed to allow modest amounts of potassium current in. Instead, after these mutations are inflicted through mistakes in the copying of DNA, these channels now let in up to 30 times more current than they're supposed to, essentially poisoning these circuits and rendering them firing uncontrollably and leading to the seizures that we see. In addition to the seizures that are the obvious manifestation of this condition, There also are profound neurodevelopmental impacts with profound neurologic impairment in for all children with this early neonatal presentation, and unfortunately early mortality, especially in the first 3 years of life. Tojo Nakayama, in work preceding our KCNQ2 work, had successfully performed a screen for KCNT1 knockdown ASOs. This inverted funnel or upside down pyramid shows that you have to go through a lot of design and a lot of screening to get to a single clinical candidate. And beginning with thousands of sequences that you computationally design, and then moving on to in vitro screening and human cell lines of hundreds of candidates, testing those for tolerability in animal models, several dozen of them, and then refining that testing using patient models of the disease, iPSC-derived neurons, and then conducting further, much more detailed safety studies that allow you to register a study with the FDA. And that work done over 2 years led us to investigational clinical candidates that I'll describe in a moment. This candidate was able to elicit 50% knockdown or inhibition of the target gene in both human cell lines and in patient neurons. We've got neuroblastoma cells on the left, patient iPSC-derived neurons in the middle, and dose response curves on the right. And we were able to do electrophysiologic recordings together with actually new Northwestern faculty, Richard Smith, as well as our colleagues, Len Kazmarek at Yale University and Imran Karashi. We were able to see that when you knock this gene down in patient neurons, you not only reduce the RNA levels, but you also correct the electrophysiologic signatures of disease. In the left are normal patient neurons grown in a dish. In red are the exaggerated currents. You see higher plateaus of those red squiggly lines that indicate that you're having much higher potassium current than typical. In blue are those same cells treated with an ASO, a random ASO that does not target the KCNT1 gene. And you see that the blue and the red lines essentially overlap. But in purple, it represents what you see when you treat these cells with our therapeutic candidate. The purple lines show normalization of currents in response to voltage steps that now looks like the normal the non-disease patient neuronal state. So that's the kind of data that we're looking for. What other sorts of data do you need to support a campaign like this? You also need to show that your compounds that work in a dish work in vivo. Now, ideally, of course, in vivo means in a patient, but before you get to that point, we use animal models to show that intrathecal, meaning spinal tap, or intracerebroventricular, meaning through the skull injections, can also have the same effects that we see in a dish. And these experiments here in mice on the left and in rats on the right show that when you look at normal levels of this gene's expression in blue, and then compare what happens to that gene after you inject our lead candidate drug, you can get reductions of the amount of gene expression by 25 to 50% via in vivo injection. You then go through safety screening, and intrathecal antisense oligonucleotides, intrathecally injected antisense oligonucleotides are not as safe as water. They're quite safe, but they can exhibit toxicities. And what's known is that at very high doses, there's a characteristic response that nearly every ASO has, in which if you give sufficiently high doses of any antisense oligonucleotide, you can render various neurologic phenotypes, including transient loss of reflexes, transient limb weakness and incoordination. And then at the highest doses, you can get sedation, seizures, and even death. And this makes the point that there's no such thing as a perfectly safe substance for anything. If too much water will kill you. These are drugs, and these are side effects that are well understood. The question is, at what doses do they occur? And do they occur at doses that give you safety margins that allow this drug to be used? Or do these side effects occur at doses that are too uncomfortably close to something that you might want to give to a patient? In addition, there's additional tests beyond this type of animal behavioral response that I won't have time to go into, but I just wanted to give you a couple of examples. And using tests like this, supporting data like I showed, we were granted permission to proceed with a very small scale trial of just 2 patients with KCNT1 epilepsy in September 2020. And I share this just so you can see what the journey could look like. We enrolled 2 patients, and we had a very careful investigational protocol where the doses are shown in the red lines in the very top of this graph. And then each of these blue dots represents some annoying investigation that we were imposing on our patient and the family to try to ensure safety and to determine whether or not the drug is working. Neurologic exams, MRIs, EEGs, and so forth. And I will share that in this example, just to finish off, in our 2 patients, we did see responses in our 2 patients, both of a good and a bad nature. In case number 1, we saw that our patient who was having between 5 and 25 seizures per day did exhibit over time a reduction in the number of seizures. Now, it's a little bit of a messy graph. Let's acknowledge that up front. But overall, there was an objective reduction in the frequency as well as the duration of seizures over that first year of treatment. I think that the case is much easier to see in patient number 2, where our patient number 2 was having between 5 and 15 seizures per day. And then at time point 0, the first dose is given, and then a second, a third, and a fourth dose in those red stars. And you see a sharp plummet in the open circles, indicating that seizures went actually down to 0. And they stayed down at 0 for almost 50 days after that 4th dose. With time, after that 4th dose, we didn't give any further doses. And you see that that effect wore off, that the seizures came back up, slowly creeping back up as the drug was being degraded in the central nervous system. Sort of a classic on-off pharmacologic effect that shows that this type of study can actually give you interpretable signals. Now, the very unfortunate news, though, is that the reason that we had to stop dosing in after that 4th dose was that we also developed a very serious side effect. Both of our patients were found to have developed enlargement of the ventricles, the fluid in the brain, that was actually very, very serious. And this is a type of condition that requires monitoring in the ICU and it requires neurosurgical intervention if the fluid builds up too quickly. In fact, one of our patients did get a VP shunt to relieve that pressure, and that stabilized her and she did well. And I'm very sad to report, but the honest truth is the first patient also had the same problem, did not have a VP shunt, and for quality of life considerations was moved to comfort care. So she did not make it. And this points to, I think, the fact that these studies are not without risk. We know now that ventricular enlargement has been seen in other ASO trials. It was seen in a trial of an ASO for Huntington's disease given to adults. And we know that now that this is a new and important toxicity of some high-dose ASOs. Now, I will say, lest I just leave just a negative message with the audience, that this is now something that once we know about it, we can monitor for it. We have protocols that allow us to detect this at the very earliest stages and avoid it by watching closely and delivering very low doses and being slower in dose escalation. So back to KCNQ2. All right, so I just described an example for how ASO gene knockdown can work with significant caveats in KCNT1. And remember that in KCNQ2, we're not just trying to knock down both copies of the gene, we're trying to knock down one copy of the gene, allele-specific ASO knockdown. As Rohan mentioned, to do this, what we do is we design ASOs that are homing in not on the generic gene target, but on a variant of genetic polymorphism, which tags the mutant copy of the gene. And one question you may be asking is, well, how scalable is a strategy like this? Well, in these sorts of campaigns, what we do is we look for genetic variants that are common in the population, and we prioritize those. For instance, consider an ASO that targets a variant that's found in 40% of the population. There are complicated equations that we use to determine how many people in the audience would actually have one copy of that polymorphism with an allele frequency of 40%. And pardon me, is that from me or is that from you? Interesting. Someone's alarm is going off.
Stop on the iPad.
On the iPad. Ah, God, excellent. All right, that's a good reminder anyway. That we're nearing the finish line. It turns out, without going through the math, that an ASO targeting a variant that's found with an allele frequency of 40%, we can predict that it should be usable in 24% of patients with KCNQ2 epilepsy. That 24% of the population would happen to have one copy of this genetic variant, and it would happen to be linked to the mutant copy. And what the take-home message from this is that this is not a one-size-fits-all strategy for sure, but neither is it a bespoke N of one. Each one of these targeting ASOs can work on a subset of the population, and to actually scale this, we need to develop a portfolio of such drugs to be able to address everybody. But the point is, we have to start somewhere. A summary of our screen, we have found 44 suitable targets in KCNQ2 based off of our studies with the Lydian Accelerator and others. 22 of these are common in the general population. We've screened at least 6 sequences for each site, but many, many more for many sites. And 17 of these sites support at least knockdown of half of the target. And 3 of these sites supported good potency and allele selectivity. But of all of these designs, another version of the slide that Rohan showed, what we found was that there were a number that looked most promising, but again, each of these, despite showing some good characteristics, some good discrimination between wild type and mutant, each of these so far has failed to pass animal tolerability testing. And so I'll just revisit what we're doing next. We are looking at more targets, finding more and better targets using long-read sequencing additional patients, considering alternative chemistries or alternative delivery methods to get around the toxicity that we're seeing, trying to work with folks in this room on better biological readouts to determine how selective is enough, and then working towards clinical trial readiness. So I think I've covered the areas that we're working. I'd be happy to engage in conversation in more detail on any of those topics. Thanks so much for your attention. I want to acknowledge the same folks as before, and then the funding for this work from the really generous support from many groups that have contributed to it. So thank you so much.
We'll do questions now?
Yeah, we'll do questions.
Okay.
With them too. They wanted to do questions together.
Yeah.
Okay.
Okay.
Beautiful work, guys. Thank you so much for your presentation. Thank you for flagging some of the difficulties and also some of the risks. I think I know the answer to this, but I wonder whether you can elaborate a little bit more about whether you think the toxicity is associated with KCNQ2 itself, or whether it's associated with the chemistry, or what are your thoughts on that?
So the good news, I think, is that so far the things that are holding us up are likely have nothing to do with KCNQ2 itself. So far, we've seen, it feels not right to describe this, but garden variety toxicity, garden variety tolerability issues that are seen with a host of other ASOs. not to be a downer, we have to be on the lookout for KCNQ2 related toxicities that could occur. And one of the issues that we talk and think a lot about is using a genetic strategy like this. Knocking down the mutant allele is great. Knocking down a little bit of the good copy is something that we worry quite a bit about. And whether that could cause toxicity is something that we're developing plans to monitor quite carefully. But we're trying to avoid that by coming up with the best drug candidate in the first place.
Questions on this side of the room?
Or on the other sides?
I had a question about the study design when you did the ASOs for the other seizure disorder. I'm assuming that the patient had to be off any seizure medication, correct? So you could monitor it.
It's a good question. So in our study design, of course, we didn't want to inhibit the standard care of our patient. And our patient was on other anti-epileptics at the time. But at the time of the trial, when the trial started, in order to manage the drug effectively, we kept their existing drug regimen the same. And then we only changed the doses of the investigational drug.
It's a wonderful talk. So I have a question. For ASOs that were used in the patients, obviously you did that and you checked them in mouse as well as rat using ICV and IT. Have you went back and looked at for example, neuroinflammation and neurodegeneration, whether the ventricles looks okay?
Yes.
Can you sort of deduce some kind of biological mechanism underlying the
Side
ventricle
effects.
problem?
Excellent question. Thanks. We have, and I'd say the work isn't yet complete, but our review confirmed that actually our mouse and rat studies were distressingly clean. Okay. Going back, we did not see any signs of inflammation, of gliosis, of microglial activation. We did not see any signs of ventricular enlargement in any of our studies, and we did studies for up to almost 5 months. So we're at a bit of a loss in our initial investigations. Now we are going back and doing a more careful look, because it is possible that ventricular changes, necropsy may not be the best way to see ventricular changes. And so we're going back and doing small animal MRIs to see if there are some sequences that can cause transient effects. So stay tuned on that. But so far, that's what we've seen.
I actually have a question for Rohan. Given that this science was all new to you 4.5 years ago, what can you say about that experience of being motivated to go and learn about all this, and what do you say to other parents about the importance of doing that?
I mean, I think the first thing I would say is like, none of this is that hard. I mean, I don't mean to diminish, a lot of work has gone into it, but I think we're so motivated for our children, we would do anything for them. So I'd encourage people to dive in. You'll surprise yourself.
There's a question up there. Maybe can I oh. Okay.
Background, you can't just.
I'm a computer scientist by training. So I was a software engineer for a while, and then I've been an entrepreneur for the last decade or so.
All right, question from a parent
Check, he
here,
really doesn't
so
have
not a
a degree in
researcher.
molecular genetics.
So my question is, as in the other study, as the effectiveness dropped off and the seizures picked back up, so if the ASO is changing the genetic structure, it's basically dropping off an allele from the cells that it particularly comes in contact with. So the drop off would come as a result of new cells not having that. So you'd have to keep dosing over time. Is that a good understanding of it?
Yeah, that's a good question. So what accounts for the drug's effect wearing off? And in many tissues, in many parts of the body, what you said would exactly be true, that new cells would be born that wouldn't be exposed to the drug. But in the brain, there's very little cell turnover. What you've got is what you've got. So it's there. It's a matter of the drug slowly breaking down over weeks and then exiting the circulation, breaking down and losing potency.
And just another question about the ASOs. How are those administered? Is it an injection? Is it just, I don't know anything outside of the names of things.
So at the moment, these are given by a needle that's and put into your lower back and injected like a spinal tap, except instead of withdrawing CSF, you're injecting drug in.
Can I also ask a question to Tim? There are these in silico prediction scores for toxicity of ASOs. To what extent do they correlate to the toxicity you saw in the mice?
That's a really good question. So our KCNQ1 drug was not the cleanest drug in the, you remember the graph I showed of it with a picture of a mouse and showing the escalating levels of toxicity? Our ASO drug was not the cleanest in that readout, but meaning that at lower doses than some, it induced weakness of the legs and so forth. However, most folks in the field can't draw a straight line between that sort of acute, temporary neurologic paralysis-like effect and hydrocephalus. The paralysis-like effect is something that happens within 3 hours and it goes away within a day. And the ventricular expansion is something that happens over a timescale of weeks. And the mechanisms are not thought to be the same. So we're still working hard on that one. We're not sure if the correlation, what a meaningful correlation would look like yet.
More questions?